Okay, hi everyone. So in the following lecture 8 and 9, we'll learn about microbial metabolism. So in these two lectures, we will learn about metabolism, anabolism and catabolism. We will also learn about enzyme and how it works. And we will touch a little bit on the oxidation and reduction and how to differentiate them. And most of the lectures will be focused on the carbohydrate catabolism, which is including glycolysis and alternative pathways, cellular respiration and fermentation. And we will also slightly touch on the protein and lipid catabolism. Okay, introduction. So in order to grow, bacteria need a source of raw material, which can be a carbon source and energy. So these sources can be same or different. For example, glucose can function both as a carbon source and energy source. And on the other hand, carbon dioxide can be a carbon source and sunlight is an energy source for a bacteria. Living things usually can't convert energy into a raw material, therefore they use the energy to assemble raw materials. Okay, let's look at the definitions. Okay, metabolism is the total of all chemical reaction take place in a living cells, which is including anabolism and catabolism. Energy is the ability to do work where it measured in the ATP. Okay, so usually living cells need ATP or energy to carry out like a three work processes, which including chemical work such as synthesis and or breakdown of complex molecules, transport work including nutrient uptake, waste elimination and ion balance, and also mechanical work, which is including internal and external movement. Okay, energy. So unit measurement of energy is ATP, or the full form is adenosine triphosphate. So it is also known as a energy currency. So ATP made of nitrogenous base, which is adenine, which is this base, and then it contains a carbohydrate in the ribose form, which is this one, and then it contains the three phosphate groups, which is this long chain. Okay, usually the high energy contains in the phosphate bonds. Energy exists in two forms. Either it can be stored energy or also known as a potential form. Such energy is stored in the chemical bonds of the carbohydrate, lipids and proteins. And the second form is the kinetic energy. It defines the energy of motion, such as energy used for transfer of substance across a membrane or enable movement. Okay, let's look at the energy production. Various species of bacteria differ in their strategies for obtaining energy. However, oxidation reduction is the main source of energy production in living organisms. So as for now, remember that oxidation is a loss of electron. For example, cells or bacteria oxidize glucose to carbon dioxide and water, and ATP will be generated. And on the other hand, reduction is gain of electron. For example, in the above reaction, oxygen is reduced and water is formed. Okay, let's look at the metabolic pathways. So the metabolic pathways can be classified into anabolism and catabolism. Anabolism means chemical reaction that require energy to synthesize complex molecules from simpler ones. For example, protein synthesis. So usually the protein is synthesized from amino acid, and the amino acid will form a peptide, and then the peptide will be formed into a protein. On the other hand, catabolism is a chemical reaction that releases energy by breaking complex molecule into simpler one. For example, hydrolysis of starch, where the starch is hydrolyzed into its monomer glucose. And just want to remember that metabolism is equivalent to catabolism plus anabolism. On the other word, energy is derived from the catabolic reaction used to derive the anabolic reaction. Okay, this is a summary of metabolism. Okay, catabolism means a larger molecule is break down into a smaller molecule, and then thus it releases energy. And this energy is used up in the anabolism to make larger molecules from a smaller molecule, and therefore ATP is a major product of catabolism, which in turn ATP is a principal substrate for anabolism, which providing the chemical energy that serves as the driving force of biosynthesis. Okay, enzymes. So enzymes are protein in nature and produced by living cells to catalyze a chemical reaction. So usually the enzyme will bind to the substrate and then to speed up the process to produce a product. So enzymes are usually very specific as which the reaction they are catalyzing, and then the rate of the enzymes are also influenced by the temperature and pH, and usually they are also very specific to particular temperature and pH. And some enzymes need additional component to show their full activity, which is known as a cofactors. And then this cofactor can be inorganic or can be organic. And the organic cofactors are also known as the coenzymes. Okay, I have a video here on enzymes, so let's watch together.
Is it odd to have a favorite protein? Well, I don't think so. Probably because my favorite protein happens to remind me of one of my favorite childhood games, Pac-Man. If you haven't played Pac-Man before, then chances are we're much older than you. But now you can play it on Google. Just Google Pac-Man. It's a Google Doodle. Anyway, I digress. But see, in Pac-Man, you have this little character. It goes around, finds these pebbles, and the pebbles fit right into it. Well, a lot of illustrations that you will find of enzymes, they happen to look, at least us, a lot like Pac-Man. I remember P for Pac-Man and P for protein. Most enzymes are proteins. Now, in the game, we mentioned there are these little pebbles that Pac-Man goes after. Well, enzymes have a specifically shaped area called an active site, where items can bind. The items are called substrates. It's very specific binding because the active site is specifically shaped for the substrate that binds there. Very specific. So what happens when a substrate binds an enzyme? Well, usually the substrate is held there with some weak bonds because it's not going to stay there forever. And something called induced fit will happen, which means the active site can change its shape even more to bind that substrate, like an enzyme substrate hug. The enzyme can either build up or break down the substrates that specifically bind to it, and we call the resulting item the product. An enzyme has the ability to really speed up reactions. Reactions that technically could happen on their own, but with the help of enzymes, they can be sped up to make processes effective for life. Let me give you a great real-life example. The enzyme lactase. Another really cool thing about enzymes is that they often end in ASE, like lactase. Many sugars, on the other hand, end in OSE, and lactose is an example of a sugar. Lactose is a disaccharide, meaning it contains two sugar molecules bound together. We don't actually digest it so well in that form. It's big. The enzyme lactase has the ability to break lactose down into smaller parts that our body can digest. And this is a lot better option than waiting for a chemical reaction with lactose to happen spontaneously. Now, with the lactase enzyme, lactose can be broken down quickly and digested. Now, there are some people that do not produce enough lactase. They can be what we call lactose intolerant, which means that if they consume foods that have lactose, such as milk, it can make them sick. They can't break the lactose down efficiently without lactase. In that example, one thing to point out, lactase, the enzyme, can break down a lot of lactose, the substrate. The lactase doesn't get used up in the reaction. It's still there. We call enzymes a catalyst because they can be used over and over and over and over in the reaction. By the way, your digestive system uses all kinds of enzymes. You have lipase, and that breaks down lipids, which are fats. You have amylase, which breaks down starch. You have protease, which breaks down proteins. So, as you can see, the digestive system is very involved with enzymes. Another thing to point out is that enzymes don't always work alone. Sometimes they get some help. Some often underappreciated but essential little helpers are called cofactors and coenzymes. They may bind to the substrate or to the active site. They help the enzyme do its job of building up or breaking down substrates into products. Now, you didn't forget our Pac-Man analogy yet, right? In the game Pac-Man, there are these ghosts. And when they touch the Pac-Man, it makes this sound. It's like, no, 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 no. And the Pac-Man shape gets all distorted in the process. So what does this have to do with enzymes? No, there aren't ghosts around, but enzymes do have certain ideal conditions that they like. For example, an enzyme that is in your stomach would have an ideal pH that is very acidic because the environment in your stomach is very acidic. Different enzymes have different ideal pH and temperature ranges. If an environment changes out of an enzyme's ideal pH or temperature range, then something that reminds me a lot of that horrible sound I tried to make can happen. The enzyme becomes denatured. That means its shape becomes distorted. It can no longer bind to its substrate. It can no longer work correctly. It is finished. Well, that's a dramatic end to enzymes.
Okay, let's look at the nutritional patterns of bacteria. So bacteria acquire energy from oxidation of organic or inorganic molecules with or without sunlight. So sunlight is function as an energy source. And then the growth of bacteria also requires some form of carbon, which is a carbon source. So generally, the growth pattern of bacteria can be divided into autotroph or heterotroph. Auto means self, hetero means other, troph means feeding. So therefore, autotroph mean is a self-feeding. So they produce their own food using sunlight as an energy source. Heterotroph or others feeding, or in other words, consumable, which use pre-formed organic compounds produced by the autotroph. And take note that human and medically important bacteria are heterotroph. And other fungus considered as a saprophyte because they live and get food from the dead host. And some human worms and viruses are considered as parasite because they live and get food from a living host. Okay, autotroph can be further divided into photoautotroph and chemoautotroph. So photoautotroph, the energy source is light, and then the carbon source is carbon dioxide. And then this photoautotroph mostly considered as a photosynthetic prokaryote, which is including cyanobacteria and plants and certain protists, which is example is algae. And chemoautotroph, where the energy source is the inorganic chemicals and the carbon source is carbon dioxide. And usually it's a certain prokaryote, for example is sulfur bolus. And next is the heterotroph. And similarly, heterotroph also can be divided into photoheterotroph, where the energy source is light and then the carbon source is organic compounds. And certain prokaryote, for example rhodobacter and chloroflexus are considered as a photoheterotroph. And the following one is the chemoautotroph, where the energy source is organic compounds and then the carbon source is also another organic compounds. And many prokaryotes or many bacteria are considered as a chemoheterotroph. And then protists, fungi, human, animals and some plants are also considered as a chemoheterotroph. Okay, let's move to metabolic pathways of energy production in bacteria. So metabolic pathway, it is a chemical reaction organized into a metabolic pathways to produce energy. So for example, glycolysis, Krebs cycle and photosynthesis are examples of metabolic pathways. And these basic metabolic pathways are similar among variety of living things regardless of their size. Or in other words, the glycolysis that happen in bacteria are similar to what is happening in humans. Okay, cellular respiration. Cellular respiration is a process cells use to convert the energy in the chemical bonds of the nutrient to ATP. In other words, the conversion of nutrients to ATP is known as a respiration process. Depending on the organism and also the availability of the oxygen, cellular respiration can be aerobic, anaerobic, or both. Aerobic is an exergonic pathway in which oxygen molecule is required. Exergonic pathway means a biochemical reaction in which energy is released. And on the other hand, anaerobic is an exergonic pathway as well, but then do not require oxygen and include anaerobic respiration and fermentation. Okay, let's look at the aerobic respiration. So aerobic respiration is an aerobic catabolism of nutrients to carbon dioxide, water and energy. And it involves release of electron as reducing power for more energy production. And most eukaryotes and prokaryotes use aerobic respiration to obtain energy from glucose. A complete aerobic respiration involves glycolysis, a transition stage, a citric acid cycle or also known as a Krebs cycle, and an electron transport chain. Now all these four stages of ATP production can be seen in aerobic and facultative anaerobic organisms. Okay, let's see the energy release cycle of glucose. So as we know, potential energy in glucose is contained in the reduced hydrocarbon bonds, right? To use this energy to the maximum, glucose must be oxidized completely to carbon dioxide and water. And the first step or the first process in the energy release cycle of glucose is transfer of glucose into the cell using a glucose transporter, which is also known as a membrane protein, which happens via the facilitated diffusion. Now after that, glycolysis will happen. The function of glycolysis is to oxidize glucose into 2-pyruvate molecule and produce reduced intermediates. And this process, glycolysis, happen in the cytoplasm of the bacteria. So this equation showing a simplified process of the glycolysis. So you provide one glucose molecule and then it will be converted into 2-pyruvic acid and then 4 ATP, 2 NADH and 2 hydrogen molecule with the use of 2 ATP and then NAD plus, which converted into 2 ADP and NADH. Okay, the following pathway is the transition pathway and also known as a transition reaction. So the transition pathways occur between glycolysis and the citric acid cycle. Before the pyruvates from glycolysis can enter the citric acid cycle, they must undergo a transition reaction in the cytoplasm of the bacteria. The 3-carbon pyruvate will undergo oxidative decarboxylation into a 2-carbon acetyl group, which is known as a 2-acetyl coenzyme A, and then it produces 2-carbon dioxide. Actually, what happens here is the 2-acetyl group is attached to the coenzyme A to form an acetyl coenzyme A. And the acetyl coenzyme A is a key precursor of the metabolite. And the reaction also produces 2 NADH and 2 hydrogen ion. Okay, the following pathway is the citric acid cycle. The citric acid cycle, also known as Krebs cycle and also known as a tricarboxylic acid cycle. It completes the oxidation of glucose by taking the pyruvates from the glycolysis by the way of transition reaction and completely breaking them down into carbon dioxide molecules, water molecules, and generating additional ATP by oxidative phosphorylation. And the citric acid cycle usually happens in the cytoplasm of the bacteria. So the 2 molecules of acetyl coenzyme A from the transition reaction will enter the citric acid cycle, and this results in the formation of 6 molecules of NADH and 2 molecules of FADH and 2 ATP. And then these NADH and the FADH molecules will then carry electrons to the electron transport system for further production of ATP by oxidative phosphorylation. Okay, the final step in aerobic respiration is called electron transport chain, or also known as a ETC. And it uses all the intermediates from the previous 3 pathways to produce a large number of ATP. And this process is happening in plasma membrane of the bacteria. So what happens is that all the intermediates from other 3 reactions, which is including NADH and FADH, are transported by a series of membrane bound protein molecules to the final electron acceptor to produce higher number of ATP. Okay, let's see the net results of aerobic respiration of one glucose molecule. So remember that one NADH will produce 3 ATP and then one FADH2 will produce 2 ATP after the ETC. Okay, if you look at the glycolysis, so where the glucose molecule will be converted into 2 pyruvates, right? And then it will produce 2 NADH and 2 ATP. And the 2 NADH will be converted into 6 ATP in ETC. In total, it produces 8 ATPs. And in the transition reaction, where the 2 pyruvates will be converted into 2 acetyl group and then 2 carbon dioxide, and it produces 2 NADH. And then these 2 NADH will be converted into 6 ATP. Therefore, it produces 6 ATP in total. On the Krebs cycle, so the 2 acetyl group will be converted into 4 carbon dioxide, and then it produces 6 NADH and then 2 FADH2, and then it produces 2 ATP. And then this 6 NADH will be converted into 18 ATP, and then 2 FADH2 will be converted into 4 ATP in the ETC. And therefore, the total ATP produced from the Krebs cycle is 24. And therefore, in conclusion, the conversion of one glucose molecule to 6 carbon dioxide will produce 38 ATP. So another thing you have to remember, one of the difference between the eukaryote and prokaryote is that in eukaryote, 2 ATPs are used to move NADH to the mitochondria for a grand total of 36 ATP from each molecules. So which means 38 minus 2 ATP which used to transport in the mitochondria, and therefore in eukaryote the total ATP is 36. Okay, that's all for today. So if you have any questions, please post in the e-learn. Thank you.